Yeah. All right, it is 9:30 and then some, and we are thrilled to have everybody here for the first time in 2 years. It is terrific to see people in 3 dimensions, but also using more than vision as a sense. Thank you for being here. I'd like to invite people to actually move lower down if you'd like. And it really, really is a pleasure to introduce the 9th Annual Symposium of the Conte Center. This is very special because we have a coalescence here of several things. One, our first in-person meeting in 2 years. Second, a focus on good news and bad news. The good news is that the focus is on depression. And as you know, UCI has just gotten a large gift that is going to be devoted to the study and the research of depression. The bad news is depression is now really an epidemic after the pandemic of the COVID-19. So we will devote quite a bit of time to that. We'll also have a chance to talk about the logistics and how this is going to work. But before we go anywhere, I think I'd like to allow 2 amazing people who have contributed so much and are contributing and supporting so much to really give the true welcome. The first is Professor Pramod Kargonekar, our Vice Chancellor for Research, and he will be followed by Professor Michael Stamos, the Dean of the School of Medicine. So Pramod, if you care to join us, thank you so much.
Well, good morning, everybody. Good morning. Good morning, good morning. Hope you are doing well. And it's a pleasure for me to be here with you to give you a very, very warm welcome to this in-person symposium after a couple of years of hiatus due to COVID-19. I think I spoke at this event maybe 2 or 3 years ago, and so it's just an honor to be here with all of you, and so good to see you all in person. I thought I would say a few things about the Conte Center and a few things about this symposium. In a few short minutes because we are already late and I think Michael is going to speak right after me. So first thing I want to say just how proud UCI and I am in having a Conte Center at UCI. There are a dozen of these across the country and we are so fortunate and you know it's no small measure due to tremendous faculty we have and the leadership of Tally that has made this possible. We had a meeting with Josh Gordon, the director of NIMH, a few months ago in connection with depression. And one of the comments he made is that these centers are really, really hard to get and even harder to keep. So that sort of underscored the importance of Conte Center at UCI. When I think about the UCI research enterprise writ large, this is one of the jewels in the crown of UCI research. So I congratulate Tally and the whole team that she leads in having this successful Conte Center at UCI. One of the things that we have really put a great deal of emphasis on is team science and team collaboration. So among other things, UCI is really fortunate to have 4 of world's absolutely leading experts in science of team science. That is to understand the process that leads to successful teams and successful research done by teams. And they come from very different disciplines, a professor in business school, 2 professors in informatics, in computer science, and a former dean of social ecology. And together they bring this diverse expertise on team science. So one thing we did was to create a team scholarship acceleration lab, which is an advisory service to faculty teams that want to work together to do team science. And so that's the theory and sort of practical advice on how to do it. But there are people who actually do it and do it really, really well. And again, Conte Center stands out as the epitome of successful team collaboration and team science. Couple of other notes. Conte Center played a leading role, and I think Tally, you will speak later today about the new grant from California initiative on precision medicine. I suspect I'm getting the name not exactly right, but it's more or less okay. And we are very excited to have the work of Conte Center have this big impact in precision medicine, supported by the California initiative. So very pleased and proud of that. The other thing that I would like to mention is the tremendous work that Tally and her leadership team did during COVID-19. So, I suspect many of you were recipients of the various rules and regulations that my office had to give out to shut down research and then ramp it back. And I can tell you that in all the meetings, Tally was there contributing and really helping Office of Research manage the COVID-19 shutdown and COVID-19 ramp up. And I think did an exceptional job. And we are very proud that in the research environment, there has been no case of virus transmission over the last 2 years. And I hope we can keep it up. We can keep it that way in the future. I want to conclude by sharing a small anecdote, which speaks to the topic of the day. So when I was at the National Science Foundation, one of the things that I was a big champion of was convergence. So the idea of having diverse disciplines come together to address major societal challenges and opportunities. And so after I came to Irvine, UCI as VCR, there was a meeting at UC San Francisco on the topic of convergence, but in the context of mental health. So they invited me. I still don't know much about mental health, but since I knew something about convergence, they wanted me to be in the meeting. And I still remember distinctly the keynote talk, which was given by Tom Insel, the former director of NIMH. And the thing that stood out to me in his talk was a claim to the following effect, that he had been director of NIMH for some 30 years? I mean, for a very long time, he was NIMH director. And he said, we spent all this money and we funded all this research. And he said he was personally really disappointed that the progress had been so little. And for a leader of a major NIH institute to say this in public was just a memorable statement. And I think what it did was to underscore for me the challenge and real difficulty in mental health, which is why I'm so excited to see the title of today's symposium, Origins of Mental Health and Disease, Research and Reality. And I hope that we can bridge the gap between research and reality, which I assume is going to be the major focus of today's symposium. So kudos to the organizers for having this theme, which also fits very well into the new Center for Depression Research that we are about to launch. As many of you may have seen in the press releases and news stories, UCI was very fortunate to receive an extremely large, one of the largest philanthropic gifts more than $50 million to create a Center for Depression Research. And there are at least 3 people in this audience whose advice and wise counsel has been critical in our initial thinking. Tally herself, David Sulzer, and Mike Yassa sitting at the back over there. Dean Stamos and I and Dean LaFerla and some other deans are working together to build that institute. So Tally, outstanding theme for your annual symposium in person. Wish much success, and once again a very, very warm welcome and a very productive symposium. Thank you very much.
I will keep my words even briefer. So really delighted to be here this morning. Thank you for the invitation, Tally, and delighted to see everybody here in person. As already mentioned by Pramod, thrilled to be here in person, thrilled to get some semblance of normalcy back. And also want to echo all the comments that Pramod made about the Conte Center and about Tally's leadership and about the faculty and learners engaged in the Conte Center. We really appreciate it and are very proud. The Conte Center really epitomizes the School of Medicine mission of discover, teach, and heal. It tackles a clinical problem by studying children and adults, takes problems that are not solvable in humans to experimental models in the laboratory, and then translates the results back to the clinic. An example of that you'll hear about later today is the California Initiative in Precision Medicine, an award that will be discussed later this morning. The Conte Center synergizes with several School of Medicine departments, as would be appropriate, right? Including, of course, the Department of Psychiatry, the Department of Anatomy and Neurobiology, the Department of Pediatrics, the Department of Neurology. But despite the fact that the center is based in the School of Medicine, it includes people from all across campus, and even from other universities, including our sister institution, UCSD. Finally, this symposium is fabulous. I had a chance to get acquainted with the keynote speaker, Diego Pizzagalli, and you're in for a real treat. But it's just one of the educational and outreach activities of the Conte Center. Other activities include going out to community schools and enhancing the UCI prestige via high-profile discoveries, papers, conferences, media interest, and really just the collection of great faculty involved with the Conte Center. So I want to thank Tally once again for her great leadership and pass the baton to her this morning for introduction of your speaker.
First, what you see now is not makeup, it's blush. So thank you very, very, very much. Which microphone are we working on? Shall we work with the leveler? Okay, well, actually, I'd like to thank our Vice Chancellor for Research and Dean Stamos for not only fantastic introductions, but actually even for staying on time, because we write on time. And it is really now my distinct pleasure to introduce our keynote speaker, Professor Diego Pizzagalli. As you can see, the theme of our symposium is the origin of mental illness from research to reality. And we're particularly concerned now about depression, because as I mentioned briefly, the impact of COVID-19 has not only been physical and societal and epidemiological, it really has been very personal. And that really has been becoming an epidemic, perhaps a pandemic, not only in the United States, but around the world. And if there's one person in this country who has taken, if you will, depression by the horns, it is Diego. So Professor Pizzagalli is a professor of psychiatry at Harvard. He actually originated in Switzerland, where he got his PhD. He then moved on to the University of Wisconsin and did a postdoc there, and then was recruited to Harvard in 2002. He rose through the ranks to associate professor, but then in 2010, he was recruited by McLean Hospital. Probably one of the smartest things that the hospital has done, together with maybe bringing in Kerry Ressler, and really created a powerhouse of the study of depression. Diego, if I may, created a center for the study of depression, anxiety, and stress. Yeah, if people wouldn't mind just be putting your phones on silence, that would be great. Thank you. And created this center, also became the director of the Neuroimaging Center, and more recently has his own Conte Center, again, that is focused on novel approaches to both the study and understanding of depression and to the use of devices and drugs to better treat it. I'm not going to tell you anymore because you're going to hear about it from his talk. I'm also not going to tell you about all the amazing work that he has done, and we know about 300 papers. I counted about 10 major awards, and we can go on forever. But again, as is absolutely known to everybody, we want to hear from him rather than me. I just think they're really privileged and fortunate to have Diego as a consultant for our Conte Center, but even more so, to have him come talk in person to all of us today and tell us about depression and how to tackle it. So let's give him a big round of applause.
Looks like it. Can you hear me? Great. What a pleasure to be here today with you. I think everybody's really excited also to be in person, and what a wonderful opportunity I have to tell you a bit more about depression. Why, of course, it's so important. And also, some of the approaches that we have taken in my lab and our center to tackle this. So actually, Tali asked me to basically, most of my talk will be an introduction to depression, etiology, some epidemiology, what do we know about this disorder? And so really the bulk of my, the first part of the talk will be on basically providing this overview and really emphasizing, the importance of course, taking a biopsychosocial models, right? Just acknowledge that depression is very multifactorial and really also require, a team science approach, if you wish. I will also emphasize that in this field, we really face a big challenge, which is the fact that, clinical, pathophysiologically, and most likely also etiologically, depression is really heterogeneous. And that's, in my view, one of the key reasons why this research has been so challenging. But then hopefully, and actually, as I go along, I'll sprinkle in a couple of findings from my lab, just to explain how we have tackled some of these issues. But then, and hopefully, I hope will be also a more optimistic message, so to speak, is that what have we been learning to tackle this huge issue, societal issue. And I'll make several points, but one will be, in my view, really important to take a cross-species approach, basically also looking across what we say, units of analysis, tackling this challenge from different angles, of course. Again, also emphasizing a team science approach. And also share with you some of our most recent work that we're actually quite excited, meaning that we have been able now to identify particular markers, behavioral, clinical, and also neural markers that perhaps might give us an idea about actually how to guide treatment selection. Basically to decide before we even start treating somebody with depression, well, given some sets of finding that I'll show you, we think we might be able to say, well, these particular patients might have a particularly good likelihood of responding to this treatment and not the other treatment. And also sharing with you some ongoing study that actually are about to start, basically, just to give an idea about how, some basic fundamental finding that emerged from my lab, but also many other labs, highlighting the role of the brain reward system, how actually we can take these findings and translate them into new treatment. And I'll finish with some final reflections. And again, I'm so incredibly grateful for the opportunity to be here with you, and it's really an honor. Okay, let's start with the first part. So, first question, what is depression? And I just recently came across this quote by William Styron, an American novelist, and I think it's actually it's very brief if you want to read it. So he was actually describing, in this book, Darkness Visible, his own experience with depression. I believe actually he experienced major depression in his 60s, or relatively late. And that's actually, how he described in this essay, what does it mean, at least for him, to have depression. And so clearly, we all know what it means to be sad, and it's obviously a very normal human emotion, but depression is not being just a bit too sad. When people meet criteria for major depressive disorders, their mood might be quite strongly, depressed, or they might be experiencing what is one of my, in quote, favorite symptoms to study, anhedonia, which refer to this profound difficulty experiencing pleasure. People basically stop doing their hobbies, stop engaging with their family, tend to withdraw, and they just describe that they just don't derive any pleasure from their life. Or they might actually have very low motivation to pursue anything that might be pleasurable. But as already nicely captured in this quote, it's much more than that. There are cognitive symptoms, difficulty concentrating, sometimes excessive guilt. There might be also, of course, suicidal ideation. And then there are also very profound vegetative symptoms. Some patients might have difficulty sleeping, might have difficulty eating. Some people might gain weight, many lose weight. There is insomnia, there is hypersomnia. So, a variety of symptoms. Now, to get a bit more formal, this is what the Diagnostic Statistical Manual here in the US, how we actually formally diagnose depression. And you make a diagnosis basically when a person meets at least 5 of these 9 symptoms over the past 2 weeks. One of these 5 has to be depressed mood or anhedonia. So these are the so-called cardinal symptoms of depression. So one of them must be present. And then, as you can see, you have a list of symptoms. Again, often you have symptoms that are opposite of each other: weight gain, weight loss, loss of sleep, sleeping too much, some people actually will start moving, talking much more slowly. Maybe loved ones start to notice that actually. Other actually becomes very fidgety. So an interesting kind of feature of this system is that if you do the math, especially because some of these symptoms can be opposite, there are many, many, many ways how people make criteria for depression. And in fact, as you can see very easily, you could have 2 people having exactly the same diagnosis and they don't have a single common symptoms. And I always, I was always kind of, I may say, bothered by this in one way, in the sense that, how can it be that, we have this heterogeneity and we as the field insist of, trying to treat everybody in the same way, so to speak, Is there any information that we can gather from behavioral assessment, from, careful clinical interview, potentially imaging that might help us, understand this heterogeneity and then harness that to hopefully either develop new treatment approaches or use this information again to guide patients to the, in quote, correct treatment for them. Okay, what do we know? I say this, very sobering fact in some respect. This was pre-pandemic. 1 out of 6 people in the US experienced depression, major depression, at least once in their lifetime. Pre-pandemic, we know it's now higher. Interesting, some of these prevalence actually particularly highest, in North America, some Western European countries. There are, pretty large cross-cultural differences or in the prevalence, so which is also quite interesting. But it has become the leading cause of disability around the world. And it basically comes, for different reasons, but because it tends to be recurrent. Again, the symptoms can be, quite pervasive. And actually, it's also because for many people actually start pretty young, during adolescence, basically, especially for girls, as you will see in a second. So that's, what makes it so such a big cause of disability. Most tragically, it accounts for about 2/3 of suicides. the incidence of suicides actually has been, again, rising pretty dramatically. Very tragically, suicide is now the second leading cause of death among young people, only after car accidents. And it also costs a lot of money to society, $210 billion in 2010. And again, this is mostly loss of productivity, of course, the cost of treating depression, but it's also, people at work not able to really function properly, not producing in some respects. So, many, many reasons. And just these are, very general facts, so to speak, but, on average, a major depressive episode might last, especially if untreated, maybe 6 months, for some people much longer, for some people, also shorter. And it tends to be a recurrent disorder, especially if people don't seek treatment, and I'll get back to this later. So studies show that about 75% will end up having more than one episode in their lifetime. And then we have this, very interesting phenomenon, which is that the prevalence is, as you might know, about double in women compared to men. And I'll get back to this in a second. So these are, again, some additional findings, epidemiological findings. These are, pretty long longitudinal studies showing that after 5 years, 40% of people that had, an index episode had a recurrence. After 10 years was 60%. After 20 years was 85%. After 30 years was 91% of them will have another episode at some times in their life. And something that really has attracted our attention and other people's attention is that for people that tend to have these recurrences, there are some interesting phenomena. One is that often the time in between episodes might get shorter. And especially, which I think is so interesting, is that, I'll show you the data, but about 80% of the very first episode that somebody has is actually immediately preceded by severe life stress. This is not being stuck in traffic, or your Zoom is not working, or other hassles that we all have throughout the day. These are, really severe life stressor, and this might be abuse, neglect, might be very, very difficult, chronic, stressful situation that are deemed uncontrollable. So 80% of the first onset of depression are immediately preceded by this type of life stress. But very interestingly, again, with people having more episodes, it seems that the link between a stressor and a future episode actually gets uncoupled, meaning that later episodes become less trigger, if you wish, from a stressor. And this begs the question whether prior episodes, especially if untreated, leave either a psychological or a neurobiological scar that basically increase the vulnerability, the risk for a future episode, even in the absence of life stress. Very interestingly, depression actually can be seen and diagnosed very reliably in preschoolers, but it's pretty rare. The prevalence of depression is relatively low, as you can see, pre-puberty. And then what happens is actually quite remarkable. As boys and girls start going through puberty, go through adolescence, but especially so for girls, you have this remarkable jump in the prevalence. And about 20% of 18 years old, or potentially even more, will have, by the time that they're 18, will have experienced a major depressive episode. And what's interesting is that this 2-to-1 ratio is actually stay constant across life, across age, so to speak. It's also constant across cultures. So despite the fact that the prevalence of depression is actually rather different across culture, the 2-to-1 ratio is actually present in all cultures. So cannot be just, some cultural or psychosocial factor. There might be something else at play here. And so, I found this, these gender differences are really important. We have currently a grant that, we are studying these, sex differences in the pathophysiology and manifestation of depression. And we just recruited our last participants last week, so we'll be able to finish the analysis. But basically, what could explain these gender differences? And as you might imagine, there are many explanations. At the beginning, especially, many people thought, well, it must be hormones. With the simple idea, you see this rise in the prevalence in girls when they start puberty. Obviously, there are quite profound hormonal changes happening. We also know that depression can occur postpartum and can also, of course, occur when women enter menopause. So there was this idea that maybe it's just about hormones, and it turned out that actually it's not really the case. There is some evidence that for women with a putative vulnerability to depression, because maybe they have a family history or a personal history of depression, when there are these major changes, giving birth, entering menopause, many of them actually are at risk for another episode. But generally, in the general population, hormones do not explain the 2-to-1 ratio. There is potentially some evidence that the timing of puberty might be actually more powerful, so to speak, meaning girls that actually mature faster and enter puberty faster compared to their peers have been found to have higher rates of depression, but the fields believe it's mostly kind of psychosocial factors, including potentially unwanted attention, the hormone cannot actually explain this 2-to-1 ratio. People have long looked whether there are potential genetic reasons, and also there the evidence is actually pretty scant, very inconsistent. There is perhaps some evidence, and again, we are working on this, that potentially biologically and psychologically, women and girls might be more susceptible or more responsive to stressor, especially interpersonal stressors. So evidence that especially when exposed to interpersonal stressors, women and girls may actually mount a stronger physiological response. And so there is an interest to see whether some of these might then be associated with increase in the risk. But there are other explanations which are also very interesting and actually quite plausible, and in fact have received quite a lot of empirical evidence, including interpersonal reasons, what Kessler and others call the cost of caring, meaning that women tendentially, on average, have a much larger social network. Social network is actually a great source of social support, but if you have a very large social network, there is also a higher likelihood that somebody that you care about might be experiencing difficulties, might be have different challenges, maybe losses, and actually this increase the chances of being also affected. Another important explanation is the trait of rumination, which basically refer to the tendency that we all have to basically dwell, sometimes a bit maladaptively, on our mood or something that has gone bad, right? And high level of rumination has been really linked to onset of depression, but also maintenance and relapse. And on average, again, I would like to stress, on average, women endorse more rumination compared to men. We all know, of course, that, and that's, it took us forever to finish this study as a small digression, this sex difference study in men and women with depression, because men, they were so difficult to find with either current or past history, because many of them had quite profound, substance abuse history. Again, other ways how they might be coping with their depression, which is why, we were really, it was so challenging to find them, that to make criteria for our study. Again, men might be coping with depression in a different way. But then, and to me, perhaps these are probably, the most powerful components is basically, social explanation. We know that, sadly, girls and women are much more likely to experience, some of these phenomena, exposed to traumatic event. We have a grant looking at childhood sexual abuse in a particular period. We have been doing this for about 10 years. So much higher likelihood of, for women and girls to be exposed to this type of horrific event. But also just generally that women in many societies are just more exposed to, to poverty, to sexual harassment, intimate partner violence, and also income inequality, or just lack of affirmation, so to speak. In many society, maybe many families, the husband, for example, might be the one making decisions about finances and so on. So, lack of access to resources and so on, that has been often linked then to higher rates of depression. And also, I'd like to just emphasize that, we know that especially abuse, again, can be a very powerful risk factor for depression. But an interesting additional twist that, again, complicate all of us research is that the timing of the abuse really matters. So these are data from my colleague, Martin Teicher at McLean Hospital, where basically over the years he has been doing research to see when some of these horrific events, sexual abuse or physical abuse or emotional abuse or neglect and so on occur, what's the probability that this will be linked to depression as a function of when the abuse occurred? And as it happened for women exposed to childhood sexual abuse, is that the peak is actually when they are between the age of 12 and 14. So this graph is basically showing the probability of developing depression as a function of when childhood sexual abuse occurred. And you might say that, in this particular case, it's especially during this age group, there is potentially a peak also here, but clearly here that childhood sexual abuse between the age of about 12 and 14 or 15 is actually what in women maximize the probability of developing depression later in life. And we've been very interested in this type of thing. So basically, the idea is that the brain is, of course, maturing. Brain region actually maturing at a different pace. Limbic regions such as the amygdala and hippocampus mature much faster compared to the prefrontal cortex. And in this particular grant that we're doing, because we're really interested in the role of the medial prefrontal cortex in regulating stress responses, we are basically recruiting women that have experienced childhood sexual abuse in that window, not before, not later, in that window, because we also have evidence that this is, again, where childhood sexual abuse actually lead to dysfunction in the medial PFC, because the medial PFC is still maturing, right? Whereas the prior 5 years of that grant, we were really focused on the striatum, on the reward system, and we knew that the striatum is actually most susceptible to insult between the age of 8 and 12. And so we were basically looking for women that reported childhood sexual abuse in that earlier stage. And in fact, as you will see in a second, we did see evidence of blunting in the reward system as a function of childhood sexual abuse. Okay, in terms of in terms of etiology, thinking first about potential biological factors, there is clearly a familiar or genetic component, if you wish. This is actually found by also studying twins that are adopted away, especially in Scandinavia. But generally speaking, people that have a first degree relative with depression, parent, for example, are a bit at threefold risk for developing depression themselves, so a bit higher than the general population. So about 40% of offspring with parents with depression will go on and develop depression themselves at some point in their life. Obviously, this says nothing about, I believe it's genetics or it's learning, modeling and so on. So, but it's just a fact. Irritability is about 37%. Clearly much lower compared to bipolar disorder, schizophrenia, or autism, for example. So, one way to read this data is that if you're not optimist, you say, well, most of the variance is actually associated with environment, which is true, right? So an environment can be changed, whereas gene cannot be changed. But clearly, as many very large studies have been done, is that there is no Mendelian transmission, right? There is no single gene that is responsible for depression, and we know now that most likely hundreds or perhaps thousands of genes that each actually explain a tiny amount of variance an increase in the risk for depression. And so the approach that we have taken and many others is actually then to basically compute this polygenic risk score, right? Just looking at the loading in hundreds of thousands of genes that have been linked to depression and see whether actually these explain changes in symptoms, changes in behavior, changes in brain function and structure, which we have been able to find. So, these are some of the genes that have been linked to depression, especially in most recent GWAS study, very large study. And, they are, you could say, they make sense, but, there is probably many genes that we have not discovered yet, and they really, really have a much larger influence. We just have not discovered them. So, with respect to basically biochemical or neurochemical dysfunction, of course, many of you, I'm sure, are very well aware that depression typically is construed as a disease associated with dysfunction in so-called monoaminergic neurotransmitter, serotonin, dopamine, norepinephrine. What I found, I don't know if people know, when I used to teach, students were always quite intrigued by this. I don't know if people know, but the first antidepressant, tricyclic, were basically developed as drugs for tuberculosis. They were not discovered as for antidepressant. They were developed to treat tuberculosis, and then they happened to see that they had antidepressants effect. And then they studied this drug, and they saw that they were basically increasing level of serotonin in the brain. And lo and behold, at that time, one of the most powerful and enduring theory in biological psychiatry was born, that evidently depression is associated with low level of serotonin. And that's why, for example, you might need to have an SSRI to treat that. And I'm all for harnessing discovery and something that you were not expecting. But I found it curious, right, that the field has been completely shaped by that discovery. And basically, with the exception of some recent exception actually, such as esketamine, for example, every single drug on the market was basically doing one thing, and it's actually increased the level of serotonin or dopamine or norepinephrine in the brain. And there are some issues with that. One, for example, is that if one of us takes an SSRI this morning, 2 hours later, your serotonin level start to rise, and yet it takes 2 to 4 weeks to see if somebody will have a beneficial effect. So clearly, raising serotonin or dopamine is not sufficient. There is something else going on. And, there might be more neuroplastic things happening, neurotropic effect that need to occur. But, just increasing this monoamine may obviously not be the whole things. They also are, moderately effective. I have a slides later on showing that actually, no matter what you give, different types of antidepressants, CBT, cognitive behavioral therapy, sleep deprivation, transcranial magnetic stimulation, all these studies show that about 50 to 55% of our patients benefit from a given treatment. And so, that's a pretty modest gain. Maybe Tom Insel, as mentioned in the introductory remark, was thinking about that, right? 55% response rate is not great. And so obviously many of us have been concerned with that, and it means that maybe some of our therapeutic armamentarium or ways are not really great, or maybe we as a field are not very good at, again, identifying patients for a given treatment. And I think that both are true. It's also interesting that I'm also a big believer of cognitive behavioral therapies, and the data show meta-analysis that CBT is actually acutely equally effective as drugs. And in fact, there is some pretty convincing evidence that CBT and similar type of evidence-based psychotherapy actually might be better at preventing future relapses, which in my view makes sense, because obviously you learn skills and that basically stay with you for your entire life. This is a busy slide, but just to show that the field actually has made quite progress identifying the pathophysiology of depression. And obviously the idea is that there might be, again, genetic factors, developmental factors, environmental pressure basically affecting this. And there are a lot of evidence that many regions, typically corticolimbic models have dominated the field, typically with the idea that the prefrontal cortex might be hypoactive in depression, might be less able to regulate emotion, regulate stress, for example. We've been very interested in the role of the striatum, the ventral striatum, what we call the brain reward system, and I'll tell you more in a second about that. But basically a variety of dysfunction, and very clearly, depression is not the disorder of one or two regions that are affected. We conceptualize it as a circuit level dysfunction, right, where networks of regions are actually are dysfunctional. And again, we have spent basically since 2002, especially focusing on the corticostriatal system, especially if you were interested in investigating anhedonia or loss of pleasure. So for example, in one of our first studies now published, well, it was in 2009, basically when we engage patients with depression, these are unmedicated individuals with depression in task in the scanner, and we ask them to, so basically they see a cue that tells them, if you're now fast in responding to a target, it is about to be presented. If you're fast enough, you receive a reward, for example, a monetary reward. In another trial, we might show a different cue and says, well, if you are not fast enough in responding to the target, you will actually lose money. And then there is a neutral condition. And basically what we found that both while they're anticipating and also receiving reward, patients with depression basically recruit less strongly different sub-region in the so-called brain reward system, regions such as the nucleus accumbens, for example, but also the caudate. And we kept seeing these findings of basically blunted activity and reactivity in the striatum to reward-related cues. And again, because we've been also very interested in studying the role of dopamine in anhedonia, especially in depression more generally, a couple of years ago we said, well, if we add a manipulation that at least momentarily increased the level of dopamine in the brain, especially in the striatum, might we be able to rescue some of this dysfunction? And what we decided to do a couple of years ago was actually, this was not a treatment study, it was a, if you wish, proof of mechanism study, in which we gave a single low doses of a D2, D3 antagonist, an antipsychotic called amisulpride, because there was some really interesting animal work and also clinical work showing that at very low doses, antipsychotics, D2, D3 antagonists, have this paradoxical effect. They actually are thought to block the presynaptic autoreceptors, and the autoreceptor is inhibitory in nature, and accordingly, you basically release this inhibition. An animal study has shown that you can very nicely increase the release of dopamine in the striatum. And in Europe, these very low doses of amisulpride have been used open label for years, for decades, to treat depression and especially anhedonia. And what we found in this particular study is that, consistent with the hypothesis, MDD subjects receiving, oh, sorry, I lost the labels, but these are the MDD subjects, these are the healthy controls. Basically, the red are patients receiving these single low doses of amisulpride, and basically they were indistinguishable from healthy controls receiving placebo. So in both the caudate and the accumbens. And as you can see, those individuals with MDD receiving the drug, one dose, were had significantly higher activation in this region compared to patients receiving placebo. Again, just a proof of mechanism showing that if you have a manipulation that supposedly increases dopaminergic signaling, we can actually rescue some of this dysfunction that we kept seeing in depression. But then, over the years, these were studied to potentially, again, emphasize the role of dopamine in depression and anhedonia, but was, quite indirect evidence. And over the years, then we also started using positron emission tomography, using tracer that are very, very good at studying, for example, the dopamine transporter. And our hypothesis was that if indeed depression and anhedonia are linked to low level of dopamine, what we probably would see if we studied the transporter is that the transporter is actually lower. Because there were many studies, especially preclinical study, showing that models that are relevant to depression, which all of them actually involve exposing animals to chronic stressor, lead to low level of dopamine and then downregulation of the transporter, right? Because the job of the transporter is to reuptake dopamine in the synaptic cleft. Because they are low level of dopamine, the brain tries to adjust that by downregulating the dopamine transporter. And that's exactly what we saw in this PET study. It was actually across different regions in the striatum, but especially in the putamen, where, consistent to our hypothesis, MDD subjects had significantly lower dopamine transporter binding compared to healthy controls. And because when we started this study, we knew that the imaging literature was really incredibly inconsistent, I think mostly because people had used tracers that were not particularly selective. We had the opportunity with one of my colleagues at McLean to basically look in a much, much more direct way in the brain. And this was actually a collaboration with Sabine Beretta, who directs the Harvard, it's basically a brain bank. And this was actually also together with Gustavo Turecki at McGill. Basically, we obtained brain tissue, especially from the striatum, especially from the putamen, where again, we had our PET finding. And we basically look at the level of the dopamine transporter, but also tyrosine hydroxylase in the putamen in individuals with depression who died sadly by suicide, and also 15 healthy controls. And this is a very busy slide, but basically the take-home message from this study was that also in the post-mortem tissue, we saw downregulation of the dopamine transporter, especially in the most mature form of these of the dopamine transporter. And there was also reduction in tyrosine hydroxylase. Now, I will say this comparison, right, as a potential confound, which is every single MDD subject in this study died by suicide, and none of the healthy controls died by suicide. They all died by natural causes or accidents. So, which is, unfortunately, a confound that is present in virtually all brain bank, but people are trying to work to counteract that. And suicide is not the same as depression, right? So there might be a confound, but we were very, we thought it was interesting that, again, two completely different modalities and approaches actually led to the same conclusion, was that the dopamine transporter is reduced in depression. And by the way, I don't personally believe that. There was also very, very nice in the PET data, there was a very nice effect. I don't have the slides with me, I cut it because of time, but there was also an inverse relationship with the number of episodes that people had in their life. That is, people with, the higher the number of lifetime episodes, the lower was the dopamine transporter binding. So potentially, it could be an accumulation effect, especially when not treated, or could be just a trait that predisposes people to chronicity and recurrences. We just don't know. Okay, I talk about stress very briefly. Again, some of this work was started by Brown and Harris in the UK, looking very carefully at clinical reports, so to speak, what happened in people's life before they had a major depressive episode. And what they found that many of them were reporting, much more so than people that did not develop depression, much higher frequency of severe life stressor. A finding that has been again replicated so many times, including in many prospective studies. So as I said, about 80% of the very first onset of depression is preceded by life stress. And so based off that, we always have been very interested in the lab to study, the role of, early adversity, for example, or also, psychosocial stressor happening in people's life. And how might this, increase the risk for depression, increase the risk for relapses, for example. And what's really interesting, if you study, this literature, people have seen, what make events particularly depressogenic. And basically, there are three ingredients. The subjects, basically perceive they are trapped. They are in a very difficult situation and they don't see a way out. Two, they feel they have no control of the situation. The stressor is completely uncontrollable. And 3, often there is a sense of humiliation. So these are, some of the ingredients that people like Kessler and George Brown and others have identified as being particularly depressogenic. And so this clearly, showed that there is certainly a very strong link between stress and depression. But people have also found the opposite, that, the opposite direction, basically, that people with depression also have a higher likelihood that they will generate, especially what we call dependent interpersonal stressors. So stressors, in some respect, they have an involvement in, right? This is not a fire approaching, or is not, a natural disaster happening. It's something that this is familiar, discord, and another type of interpersonal stressor. So certainly there is also this another direction. And so we, again, years ago, really got very interested in studying this. So why is it that later episodes of depression become more uncoupled from stress? And a testable hypothesis was that perhaps a higher number of episodes is associated with dysregulation in brain regions which are really important for regulating stress. And the 2 most kind of prototypical are the hippocampus and the medial PFC, because these are the regions that basically are in our brain to shut down stress responses, right? Amygdala and other regions excite the stress response. Hippocampus and the medial PFC, among others, are really important for shutting down stress responses. And lo and behold, when we did morphometric analysis in hundreds of patients, this is basically looking at cortical thickness and also volume in subcortical regions. These are the regions that basically, the only region that survived, a very straight statistical threshold, and was the medial PFC and the hippocampus. And there was an inverse relationship. So with higher number of episodes, the higher the number of episodes, the smaller the cortical thickness or the volume in this region. And so, that was interesting to us. And also, I should say that in this particular study and others that we have done, we have seen that people actually with higher number of episodes, just in the month before the scan, happen to report lower level of stress, perceived stress, which is, again, seems consistent with this hypothesis that maybe there are neurobiological sequelae, especially from untreated depression, that basically increase the risk for depression, and depression can actually arise even outside the context of life stress. And to give you, again, also some good news, so to speak, is that treatment, for example, successful treatment has been found to reverse some of these morphometrical findings in the hippocampus. This is a really nice study done in Germany by Frodel and co-workers, showing that successful treatment was actually associated with normalization of hippocampal volume, both the left and the right hippocampus. So again, treatment really matters, not only in terms of obviously alleviating these profound symptoms, but literally, in this case, reshaping the brain, if you wish. So, as I said, we've been very interested in dysfunction in this mesolimbic system, right, which originate from the ventral tegmental area, substantia nigra, project to the nucleus accumbens, and the ventral striatum accumbens, and dorsal striatum, the caudate and the putamen, for example, and then project up. We know from decades of work in non-human primates and in rodents, this system is incredibly important for reinforcement learning that specific action, for example, lead to reward. But it's also very important for incentive motivation. And again, I've always been a pretty avid reader of preclinical literature, and I was really intrigued by the fact that lots of manipulation that people do to animals, they basically all have a common ingredient, which is often exposure to something chronic that the animal cannot control, right? Could be chronic mild stress, could be chronic restraint, could be material separation, or other manipulation that have been championed here at UCI, and I'll get back to that in a second. So they all have this ingredient that the animal cannot cope, basically successfully cope with the stressor, and this actually has led to downregulation of this system. And that's why, as I said, early on in the first 5 years of that particular R01, we're really focused on studying the role of the striatum in basically the consequences of sexual abuse. And this particular study was actually a first study we did in actually a relatively small sample, but was a really precious sample because this individual had been followed for 20 years. And their abuse actually had been corroborated by social services. And so there was a lot of information about that. And when we brought them back, they were in their 20s. Remarkably, just one of them was meeting criteria for depression. Others had psychopathology early in life, but just one had met criteria for depression. Just one was on medication. And yet, 20 years after the abuse, they were still less able to recruit the brain reward system, particularly or specifically when anticipating a potential reward. So there was no difference for no incentive or even the loss condition. So suggesting that potentially exposure to very, horrific events in childhood, again, might leave a scar and actually really leave a pretty hypofunctional reward system. Later on, together with a postdoc, we kind of expanded this. This is a complicated slide, and I think I'm running slightly behind. So just to say that, we also engage patients in tasks where they have to learn by trial and error to find out which stimulus is basically associated with more reward and which stimuli actually are associated with more punishments. So they win points or money for doing so. And what we found in this particular study, this was in women with actually a past history of depression. They had no symptoms right now, but they had, again, experienced childhood sexual abuse. And basically, the take-home message from that study was that compared to both the healthy controls and the women with past history of depression, but no CSA, women with CSA and MDD, past MDD, were basically less able to learn from rewards. And was actually also here quite specific to reward. They had no issues basically identifying the B stimulus, which was the stimulus associated with more punishments. Again, highlighting potentially disruption in the brain reward system. And fortunately for us, we had, again, the good fortune of collaborating with several great faculty here at UCI, Tali Boran, but also Dr. Stern and Annabelle Short. And basically, we used the paradigm that Tali had really developed and optimized, basically where animals are basically exposed to a less amount of bad and nasty material, so to speak. And what Tali and her team had found was basically animals exposed to this manipulation actually grow up to show an anhedonic phenotypes, right? Basically, and other neurobiological abnormality. And so in two different cohorts of rats that were basically bred here at UCI, and then Tali and her team sent us to us at McLean, we engaged them in a task, which I'll explain also in a second, but basically it's a task where we train animal to basically decide using touchscreen technology whether a short or a long line had been presented. And first we trained the animal to associate, let's say, the long line to this left virtual lever and the short line with this right virtual lever. The animal actually can learn that in 15 to 20 days. And then what we do critically is that when they do the test, we disproportionately reward identification for one stimulus over the other. So the animal is not really sure what is presented, but we actually shape their behavior toward one stimulus by giving more reward for one stimulus over the other. And incidentally, we basically back-translated this from a version that we've used for about 15 years in humans, where humans were doing the same. They had to identify whether the mouth that was flashed for very briefly on the screen was long or short. And you see actually an example of one of these rats rearing up and using this touchscreen technology to make decision whether this was the short or the long mouth line. And what we found, again, in two cohorts that were basically studied about a year apart, because something like a pandemic also happened, we were just blown away. We had never seen such a reliable and strong effect. The effect sizes were very, very large. So this creature that had been exposed very young in age, you see it here, they basically were remarkably blunted in their ability to develop a response bias toward the stimulus paired with more reward. We started small, then Tali and her team sent us many more rats. These rats, actually, because of the pandemic, had to be on the shelf for about 45 days or so in quarantine when they came to McLean. And we're just blown away that they still had these profoundly anhedonic phenotypes, inability to learn from reward, because they had been exposed to the pretty short but very powerful manipulation. Again, showing that exposure to early adversity might affect the reward system. And now we have collected the brains of this second cohort, and we're basically looking at potential molecular marker that may be associated with that. Tali, how am I doing with time? 5 minutes. I'll skip forward, actually. And basically, again, summarizing this part of the talk, basically, what I wanted to convey is that, needless to say, depression is complex, but it's very evident that there is, a shared biological vulnerability. We think that especially potentiation in stress responses might be an important component. But then when people are exposed to stress, especially those that have this vulnerability or diathesis, actually might obviously respond much more forcefully, and there might be a cascade of neurobiological and psychological consequences that then in turn can interact with many other variables. For example, exposure to inequity or discrimination, poverty or bullying, for example, and other obviously psychosocial factors such as social support, typically, which is identified as a protective factor. So, obviously a very complex model that highlight both psychological, biological, and social factors. So then maybe a bit briefly. So in terms of the second part of the talk, just to show you also how we might be able to tackle some of these challenges, right? So one basic, my lab, generally speaking, has been very interested in answering some of these questions, right? So can we use a translational approach to basically try to predict, if you wish, the future? And the future could be the first onset of depression in youth at risk for depression. It could be actually predicting future symptoms or potential relapse in people, adults that have had depression, or actually, again, predicting treatment response, right? So that we might be able to move closer to personalized treatments. And as a representative example of the first line of work, again, we have been studying young children, completely symptom-free, but they happen, some of them happen to have parents with depression, but the child has never had depression. And what we did was to bring them to the lab. You note that we selected 12 to 14 years old because, again, it's before the surge, so to speak, right? And basically, we did the electrophysiological, EEG, MRI, MRS assessment, a bunch of behavioral assessment, clinical assessment, and we have been following them over the years. And these are interim findings published a couple of years ago when the sample was much smaller. And basically, by just looking at the volume of this, what I've been calling the brain reward system, in this case, the putamen, we found that in these youth that have never had depression themselves, the volume of the putamen was actually negatively associated with their future anhedonic symptoms while controlling for current anhedonia. So basically, youth that had a smaller volume in this brain reward related region actually were the one that reported more anhedonia 3 months later. And again, now we have many more subjects, and we are about to also look at 2 years follow-up clinical data to see whether we can identify markers that predict first onset of depression. I maybe skip this one here. This was a study where basically we were able to look at the identify markers that predicted recurrence of symptoms in adults that were symptom-free when we tested them, but happened to have a past history of depression. And the take-home message from this study is that we brought them into the lab, we engaged them in a cognitive control task, computerized task to measure basically their prefrontal cortex function, and we did so before and after stress. And in the middle, we exposed them to a psychosocial stressor, which is basically, exposing people. I can tell you more in the Q&A, but believe me, it's a well-validated psychosocial stressor in the lab to basically increase cortisol quite strongly. I can give you all the details. And basically what we found is that, very interestingly, pre-stress, there were no differences behaviorally or electrophysiologically between remitted and healthy controls. But when we exposed them to stress, the remitted sample was actually less able to recruit the prefrontal cortex in a cognitive control task. We use a flanker test for that. So again, raising the possibility that people with a history of depression might be less able to regulate stress and maybe recruit prefrontal cortex, which is very important for regulating emotion and stress. And that might be one of the ingredients to explain future episodes. And in fact, in this particular study, we found that the greater the reduction in the prefrontal cortex recruitment after stress, the higher the depressive symptoms 18 months later. But I leave you with, boy, I really underestimated how I would speak. So this was actually the, I'll finish with these sets of studies, which is basically, again, going back, whether we can find markers that might predict response to treatment. And the backdrop for this study was the idea that we see this pretty modest response, right, to different types of intervention, and we wanted to see whether we can identify markers that predict whether people will respond to treatment. And basically, what we found was in this very large study, patients were randomized either to an SSRI or to placebo, and if they failed to respond to the SSRI, they were switched to bupropion, which is an antidepressant with a very different mechanism of action. And basically, we were interested in seeing whether we can find any predictors that basically identify people that fail sertraline but then go on to respond to bupropion. And we then looked at their pre-treatment data to identify such markers. And the take-home message of this is that, I'll tell you, without maybe explaining the slides, it is that quite interestingly, and we are still trying to fully understand this, patients that were depressed at week 0, so to speak, they were depressed, similar to the other patients, but happened to have a slightly more normative ability to learn from reward, and they also had a more normative connectivity between the accumbens and the ventral medial PFC, ended up failing SSRI, but then doing really, really well on Wellbutrin. As if, a bit more responsive brain reward system is actually a prerequisite for responding to a drug that, increased dopamine. And we're very intrigued by this because basically now what we're doing prospectively in a recent grant that we received is to use this type of marker to decide, how to treat patients. So we bring them to the lab, do an MRI, do some of our behavioral testing, and basically immediately analyze the data and decide, okay, this person should be treated with an SSRI. No, this person needs to be treated with bupropion, and I'm happy to tell you more. So I'll, can I take one more minute, Tali? This is basically what we're doing. As Tali mentioned, we have our own Conte Center to study basically potential novel targets for depression, and I'm happy to tell you more about that. One of the upshots is that we're studying this again across species. This is what I just described, this smart trial, so to speak, where we will prospectively assign patient to treatment. We are also studying other type of treatments such as ketamine that have been found to have very, very rapid antidepressants effect. We want to understand what might predispose to this response. And actually, this is an example that we're quite excited about. I've been telling you all this finding, highlighting the role of blunted brain reward responses in depression, we are teamed up with a group that has developed a very clever virtual reality-based fMRI-guided neurofeedback, basically with the goal of increasing nucleus accumbens activity. We continue our work looking at the effect of early adversity, but really actually focusing on oxidative stress and inflammatory pathways. And we want to really expand some of, again, our cross-species work now to more, models relevant to depression, and I'm happy to tell you more about that. If you want to read more about this, there was this really very interesting paper that came out in Lancet a couple of weeks ago that provided a really important review of depression. And this is my last slide. Obviously, tackling depression is very challenging, but I do think, I don't want to leave you, without an optimistic message, so to speak, that we think, I personally believe that we are at a crucial step, so to speak, where we have the technological tools to really try to make a difference in people's life. And these are, some of the things that I think will be really key if you want to make a difference. There are many challenges. I used to say when, maybe I was interviewed by the press, yeah, we just found this cool finding. And, I think maybe in 10 years, this might become, useful for patients. I think, I know our patients don't have 10 years. We need to really accelerate this. And really the way how we do science will be important. So I'll stop here and thank you for giving me a couple of extra minutes.
Diego, thank you for a real tour de force. This is remarkable. I also would like to take the opportunity and to welcome and thank the people who are watching this remotely. And what we can do is one of two things. We either can take questions and answers now, or we can have the next talk and then have a general discussion and perhaps stay on time in that way. And perhaps we'll do that. We will actually give you a very different orthogonal example of how to go from, if you will, research the lab to reality, and then have a nice long question and answer that will be applied to both. So again, those who are remote, I think there's a way to use the chat, and if not, text James. And I'd like to also use the opportunity to give just a little bit of an idea about how today will run. Again, we launched the keynote speaker to allow both people in the audience and remote, those who are virtual, to join. I'd like to encourage everybody to move forward in this particular venue, if you will. And what we'll do is we'll launch now to the next talk. We'll then have a discussion. Before noon, we will have a break. Then there will be the posters with poster tours. So everyone who's got a poster will actually have a chance to give a 2-minute blast, if you will, or elevator talk, or Beckman Center talk, or Conte Center talk, whichever you like. And what we'll do is we have more than 40 posters. We will divide into 4 different tours. So everybody in the audience will be able to pick and choose their favorite theme and go join a particular poster tour, or we'll be able to just switch around. Also, you can linger around the posters as you wish. We'll then have lunch, and looks like the authorities upstairs have indulged us, and we'll have a wonderful weather. Then we will move to this afternoon session that will be run by Mike Yasa, and we will go into Data Blitz. These are people who were chosen from among the posters, really to highlight what the, if you will, best generation of neuroscientists and in other disciplines is doing to address issues of early life adversity and mental health. We'll then have 3 short talks from recipients of the Conte Center seed grants, and I encourage you, if you haven't, to look into that and consider submitting one. And then we'll end. So that is sort of the overview of what we're going to be doing next. Are people comfortable with going for the session, for the question session after the next talk? All right. So with this in mind, I would like to invite Professor Laura Glenn to the podium with me. Dr. Laura Glynn is a professor at Chapman University and got more titles than I can even care to admit here. She is a co-PI of the California Initiative Award and will present some of what I'm going to share with you in the next 45 minutes. And that will then take us to the next set of slides. If I can ask IT to put on the next talk. That is fantastic. Okay. So again, the theme of this symposium is the origin of mental health and illness from research to reality. I think you got an amazing overview of the scope and depth of one of the major mental illnesses, which is depression. And the question that Professor Pizzagalli left off with is, what do we do about it? How do we even get there? Well, what Laura and I would like to tell you is how folks from the Conte Center, working with the community and using a variety of methodologies, really aim to bridge the gap. So let's launch right in. So what I'd like to tell you about is the California initiative to employ precision medicine to address the health impacts of adverse childhood experience, again, through a collaborative precision medicine approach. So what am I talking about? Well, we owe this brilliant and amazing and visionary notion to two people, to Dr. Nadine Burke-Harris, who is until recently the California Surgeon General and has devoted her life, passion, career, and resources to really address early life adversities, or ACEs, in many different ways, and I'll talk about this in a minute. And to Dr. Julianne McCall, who's the co-director of precision medicine in the Office of Planning and Research of the Governor of California. We are fortunate to have Dr. McCall with us today in the audience, and I'd like to ask Dr. McCall to stand up so we can acknowledge her. Julianne. Thank you so much for actually surprising us at the last minute and actually coming. You couldn't have done anything nicer. So Doctors Burke-Harris and McCall came up with a quest, an RFP, a request for proposal, to really address health impacts of adverse childhood experience through a collaborative precision medicine approach. When we at the Conte Center, all of us, the team of psychologists, neuroimagers, psychiatrists, basic scientists, when we saw that, we were elated, because this is an opportunity for us to really see if our science, if what we're discovering is translatable to the real world, if it can actually take us from the lab to reality. Because what we've been focusing on in the Conte Center is really a very simple question. What determines emotional and cognitive health and disease? What determines how well we learn and laugh and cope during childhood and throughout life? And as Dr. Pizzagalli has shown us, of course, genetics plays a very key role in that. But environment, particularly environment during development, clearly is quite important. In fact, as you've heard from Diego, early life adversity, defined as poverty, trauma, stress, abuse, is strongly associated with lifelong risk for mental health problems and cognitive function problems later in life. But as Diego also showed, what we can do in human research is really correlation. And can we understand deeply the causal relation of genetics and environment and development? Again, just to give you a very simple example, if a child of a depressed mother is more likely to become depressed too, what's the mechanism? That mother, of course, will have a depressed behavior, and that maybe influence the child. But that mother will also be accountable for 50% of the child's gene. How do you sort this out? So what we've looked at in the Conte Center is really a question of what is early life adversity, what's important, what are the parameters that really shape outcome, and as you've seen from Diego, this is not easy. What are the outcomes, both associations in human and causal in animal models, and what are the mechanisms? Because again, we need to understand the mechanism at the level of imaging, at the level of molecules, at the level of circuits, to be able then to turn back and really change that. So we began to think about those things, of course, adversity, poverty, lack of resources, violence, war, migration, parent absence, neglect, abuse, maternal depression. We know all these. But surprisingly, rigorous studies and the newer studies are actually suggesting that whereas established types of early life adversity explain wonderfully population level risk levels, they don't do quite as well to explain that at the individual child. And maybe I'll stop here for a couple of minutes, seconds, just to let you look at this article that I strongly recommend, because what they conclude is that ACEs scores, the level of early life adversity as measured in the state of California and elsewhere, forecast very well mean group differences in health. They don't do quite as well in explaining an individual outcome. So this is a pretty strong statement, and let me just show you what I'm talking about. So at the group level, the article looks at 3 major studies around the world that look at the health, the risk of having 0 or 1 or 2 or 3, 4 early life adversities that we talked about now, lack of resources, neglect, abuse, violence, what have you, and looks at any mental health problem, and the correlation is fantastic, okay? However, when you look at an individual, you see something like that. Not going to go into the nitty-gritty details, but this is specificity and sensitivity. And if you have just a chance relation, you're pretty much going to have in this rock and the receiver operating codes, you're going to have something that looks like the purple line. What you really aim for is something, something that will give you a curve that looks like that. So at the individual level, the power of the various aggregate and individual types of early life adversity is not quite as good. But this is really what we need. We need to know if 2 youngsters who have had the same type of early life experiences are at risk, at equal risk, or we'd like to predict if one is going to be just fine, but the other one may not be. So there's something that's missing here. So what is missing? Well, one of the things that became apparent to us, and has been apparent for the past century or so, is early in life, it is the signals from the parent, the caretaker, the proximate environment, if you will, that really buffers anything that the environment throws at you. You can live in a small village in Nigeria, or you can live in Beverly Hills, California. What you're going to see as an infant, as a 1 or 2 year old child, and definitely prenatally, is the environment that comes in from parent, mother, father, what have you. And so that we began to consider that if we want to understand the causality and the mechanism of how early life adversity influences mental health risk, we need to really look at this categorically. So again, whatever the signals are, including early life adversity, that will reach this child's brain will be both direct, as we see here in this very poignant picture, but also will influence the behavior of the caretaker, the parent, the mother, and that will also be a signal or set of signals that will influence the child. So that if we want to generate animal models, and Diego talked a little bit about the animal model that we use here, which is simply a simulated poverty, a lack of resources, we need to look also not only at the physical environment, and by the way, we know now that the physical environment contributes relatively little to, quote unquote, the mental health outcome or the cognitive function of these rodents when they grow up. It really is the signals from the dam, in this case, in the cage. And we began to look at these signals. This is a busy slide, and the bottom line is there is no difference in any measure that we looked at typically, which is the total duration or the type of nurturing, caring behaviors that the mother has. But when we look in live, when we look in real time of what's going on, we saw some pretty potent differences. Is anybody in the room here who does not see a difference between the behavior of the mouse on top and the mouse on bottom? Okay, so what are we looking at here? This mother mouse will eventually settle down, and the total duration of care and type of nurturing behaviors that she will provide the pups is the same. But one thing that was very clear to you is the duration of each bout is really, really short. In essence, and this is a brilliant slide that Laura put together, we see total fragmentation of the behavior. In addition, working with Professor Hal Stern and his team on trying to look at the sequences of these events, we found that they were also quite different. So again, this is simply a probability matrix of what's the probability that behavior A will follow behavior B, namely, they will be predictable. And you can see here, after behavior C, there's a very high probability that behavior O will happen, and you'll hear more about it from Laura. On the other hand, after behavior N, behavior C never shows up, 0 versus 0.8. Whereas in the limited bedding and nesting cage, everything is blue and green, namely, very unlikely. So in the rodents, we actually have both fragmented and unpredictable set of sensory signals that impinge the barrage of the developing brain. And we know now from mechanistic experiments in animal models that they seem to interfere with the normal creation of synapses or maintenance and pruning of synapses and generation of normal brain circuits. Now, the outcome of that is important, at least in rodents, because I'm not going to read the whole list, but fundamentally, there are memory and cognitive problems, altered sensitivity to subsequent stress, with appropriate brain region neuronal circuits being disrupted, and also serious, quote-unquote, emotional-like problems in rodents that are highly sex-dependent. And this happens after just 1 week during a sensitive period early in life of being in this unpredictable, fragmented, chaotic environment. You don't see it if you only have situations where you separate the mothers or whatever. So this seems to be really something that has to do. And this is all nice and dandy. So in rodents, we know that unpredictable signals are important. But who really cares about the mental health of rodents? All yours, Laura.
Next, I'm going to talk a little bit about the relevance of this idea in the context of humans. And before I talk to you about how we approach this, I do, the Conte Center, as we've been discussing already, is team science, I think, at its best. And I would have to acknowledge, there are so many people even in this room that both Tally and I could acknowledge, but in terms of translation of this rodent paradigm into human work, I really want to call out Alicia Davis, who's here, and Hal Stern, because they were the leaders in this collaboration. So how are we doing this in humans? In my labs and Alicia's lab, we bring mothers and their infants in at 6 and 12 months of age. We give them a standardized set of toys to play with, and we videotape those interactions. We then go on to code in real time what we have characterized as the relevant sensory signals in these human mothers. And what those look like, we're coding along 3 channels: auditory, tactile, and visual. You can see here some of the behaviors we're coding. This is an example of a time series of those behaviors across this 10-minute play period in a single mother, so you can see how they're distributed. The next thing we want to do is characterize the transition probabilities between these different sets of sensory signals. And so what we can do here is put these transition counts in a matrix, and you can see on the column or the rows, we have the from action, and on the columns, the to action. And you can see, for example, in the 65 highlighted here, what that means is that in the context of this interaction, a visual sensory signal was followed 65 times by an auditory visual signal. We then translate these counts into probabilities, and then I, as the collaborator with Dr. Stern, wave my hands, and we say, oh, now we have an entropy rate, which is what we call a quantitative measure of the degree to which a future behavior can be predicted from a current behavior. So this is our measure of unpredictable sensory signals. And we're able to characterize this again in both humans and rodents in a similar way, obviously with different sets of behaviors in the rodents. And what you can see on the left is that in our human cohorts, this results in a sort of naturally occurring distribution of these unpredictability rates. In the rodents, because we're manipulating this experimentally, as Tally showed you, we get nice group separation. But it's worth noting that you can look at that distribution in the humans and see we have nice variability. Those are data from one of our human cohorts. After characterizing this, we then can go on to ask questions about how these unpredictable sensory signals might link to neurodevelopment in both species. And in this first paper, we looked at memory performance in both the rats and 7-year-old children. And what we saw was, in both cases, higher levels of exposure to these unpredictable sensory signals resulted in poorer memory performance. This is a recall memory task. And I want to note that in the humans, we pay special attention to possible third variable explanations, because of course, if we've been discussing, we can only do correlational studies in those groups. But we have pretty thoroughly probed all of the sort of alternative explanations we can think of. And I can tell you that in the cognitive data I'm going to be showing you in humans, that we know that these associations exist beyond the effects of maternal sensitivity. So we code it in a more traditional way from these interaction videos we have, child age, maternal depressive symptoms, and socioeconomic status, among other things. So we've pretty carefully sort of tried to triangulate in on the unique effects of this novel early life exposure, unpredictable sensory signals. Since then, we've continued in both the rodent and human work, but I'm going to talk a little bit about one domain where, if I think, and Alicia, you can wave at me if you think I'm saying something wrong here. I think one of the most robust, consistent finding or cognitive outcome we have in terms of these unpredictable exposures in the humans is effortful control or self-regulation characteristics, which is really important because we all know that effortful control is a sort of transdiagnostic predictor for later psychopathology. What you can see on the left, for example, is that in 1-year-olds, both in one of our California cohorts and in a cohort of collaborators in Finland, you can see that higher levels of unpredictable sensory signals are predicting poor effortful control at 1 year of age in both cohorts. On the right, what you can see is that association is persisting through 9 years of age in these kids. And more recently, in some unpublished data, we know that it's persisting at least into adolescence. So in this cohort you see here, those kids are, some of them young adults now. In a younger cohort developed as part of the Conte Center, we've looked at the association between unpredictable sensory signals and effortful control in this classic snack delay task. I did do the analyses with continuous predictors and outcomes, but to visualize the data for you, what I have here are the top and bottom quartiles of exposure to unpredictable signals, and you can pretty clearly see that the kids who had been exposed to higher levels of unpredictable maternal signals are waiting on average about half the amount of time as the kids who had fewer exposures or lower levels of exposures. And I think it's time for the next component of the Laura and Tally show. You'll see me back, but she's going to take over for a moment.
Thank you. So the amazing findings from the, if you will, the human portion, which is the major one in the Conte Center, including projects by Laura and Alicia with input from Mike Yasa and Hal Stern and all of the amazing trainees, students, collaborators, and affiliates that go with it. We thought that, gee whiz, it looks like, in addition to everything that we knew about what early life adversity is, there may be something that may explain some of the variants. Maybe, indeed, unpredictable signals from parents and environment are important. And again, you kind of ask, well, why should they be important? And again, in rodent models, we can see now that you do need to have appropriate sequences of signals that will really allow neurons to fire together so they can wire together. And this is work that is now being done in, if you will, Project 1 of the Conte Center in the animal world, in the mechanistic studies. And some of the posters you'll see this afternoon will really highlight that. So the question is, this is great. It may be working in cohorts that we have in the lab, in the clinic. It's nice to know that it's not only in Southern California, it's in Finland. And by the way, some of the instruments that Laura developed are now translated into Spanish and Finnish and German. What else? I think there's another one. Dutch, right. And it's used around the world. But is this really important in the real world? Is it really important for children in Southern California and in California in general? And most importantly, can we do something about it? And this was exactly where we were when we saw this amazing request for proposals in 2019. Actually, we showed you some data that we gathered after that, but this is sort of the mind frame. And again, the California Initiative to Advance Precision Medicine will award a certain amount of grants to really try to address, for a given child, which is the one thing that's missing, as you've seen, the ability to predict outcome so that we can actually intervene early. And also, we thought we would like maybe to consider to find a predictive marker. So I'm not reading what's on the slide here. Take your time, do that yourself. So the idea really was to leverage the amazing individuals in the Conte Center and in other entities to work on this together. And when we talk about team science, it's not only just people coming here. I'd like to thank Marcella Wood for the slide, working on the same thing, but also reaching out to the community. We're working together with the communities in Southern California through the help of Dan Cooper and Candace Taylor Lucas, and the work that Laura is doing with her team to really reach out the community and work with them on how to conduct the study. But if we are really dealing with a novel, actionable, preventable ACE, we need to do something about it. Because think about it, as much as we would like, it is a serious challenge to eliminate poverty, neglect, abuse, war, migration. But we can teach individuals about some aspects of predictable, consistent signals. So we elected to come up with a proposal. Again, identifying these two unmet needs. Is unpredictability important in the real world? Does it really contribute to the variance, which is something that we see in controlled cohorts in many, many youngsters in Southern California? And again, the other aim is, can we predict with the precision of an individual child that doesn't seem to be as well possible when we look at regular quote-unquote ACEs or well-established ACEs. So the project plan involved two aims. Now, do I give it to you now, Laura? Okay, well, establish the impact of early life unpredictability and novel actionable ACE on neurodevelopmental outcome in 100,000 kids in Southern California. And aim two is to capitalize on an innovative strategy that we tested initially in rodents to find a predictable marker. We put this, of course, in the context of the roadmap of resilience that Dr. Nadine Burke-Harris has established in California. And that particular roadmap involved every pediatrician in California administering a questionnaire for ACEs, for adverse childhood events, so that we knew that in the clinics in Southern California that we're going to be working with, this will be the case. And with this in mind, we reached out not only to existing collaboration in Chapman, but also to Children's Hospital of Orange County, and I will give credit to these individuals at the end, and came up with a plan. Back to you.
So 100,000 children sounds like a lot. I'm sure some of you took a pause at that. So how are we going to do that? So aim one is really to look in a large group of children at the question of whether unpredictability is predicting neurodevelopment, and not just neurodevelopment, as I'll tell you, we're looking more broadly at both indicators of both mental and physical health. And what are we doing? As Tally mentioned, we were really fortunate, and one of the reasons I think we were successful probably with this proposal is that CHOC was an early implementer of this idea of screening for adverse childhood experiences, and in their pediatric primary care clinics, they had already rolled out this screening. So at every well child visit, for every kid in these clinics, they're already doing a screening for adverse childhood experiences, and that would be the PEARLS, which is a validated instrument that they're using. And just as a point of reference for those that don't know, CHOC treats upwards of 70% of the kids in Orange County. So this is a very representative sample of kids who's being screened. So our hope was that we would leverage that ongoing existing screening by adding an additional short screen to target the idea of unpredictability and exposures to unpredictability in childhood. Fortunately, a few years also before this RFP came out, the Conte Center as a group came to the realization that although we increasingly see the importance of these unpredictable sensory signals and experiences in early life, it's not feasible in many research settings, and certainly not in clinical settings, to do these sorts of intensive prospective assessments of what's going on in early life. And so what we did was develop the QUIC, the Questionnaire of Unpredictability in Childhood, which assesses via self-report exposures in social, physical, and emotional domains in childhood related to unpredictability. In that initial validation paper, which was a center collaboration, we showed that the QUIC has very good psychometric properties and that it is a good predictor of depressive symptoms, anhedonia, and anxiety across 3 cohorts we looked at. So adult women, male and female adolescents, and adult male veterans. And again, just to highlight this idea about the unpredictability being a sort of independent novel predictor, we did test to the extent that we could in these cohorts whether it was independent of things like childhood trauma, childhood SES, and the answer is that it was independently predictive from these other known ACEs. So even a 38-item questionnaire is a lot to ask in a clinical setting and research settings where time is short. So our next step was to develop a quick version of the QUIC 5. There's a poster about this here today if anybody wants to see it. And what I can tell you about this is these 5 items across those 3 cohorts are correlated about over 0.85 in each cohort with the original 38-item scale, so very nice concordance between the short version and the long version. And in terms of its predictability for those mental health outcomes that I showed you, the effect sizes are virtually indistinguishable. So this is really exciting because I think we have a short screener we can use in the context of clinical and other research settings. So in terms of where we are, this project started in this past summer. Implementation of the screening has been an uphill climb, as it always is, dealing in clinical care settings and collaborating with our team as good as they are. I want to do a shout out to my team in Chapman for their unwavering persistence in getting this rollout going. We are currently now fully up and running in 7 of the primary care clinics. They are screening at every well child visit along with the pearls. And I can tell you, as of this morning, though things, we had some kinks to work out, but we already have in our hands 1,500 completed screens, and those are coming in by the hundreds every week now. So our next steps are to continue the rollout. There are another next 7 or 8 that we've been in conversation with, and we'll continue to roll out into those clinics. And it's also the case that my group has been working closely with the data analytics group at CHOC, because one of our next steps will be to connect the PEARLS scores, the quick scores, and a wide range of child mental and physical health outcomes that we'll be able to get from medical record abstraction from these kids' pediatric records. Before I turn it over to talk to Tally or have Tally tell you a little bit more about the second aim, I did want to just emphasize or discuss a little bit a really important component of this project, which is really a cross-cutting theme. It really involves both aims, and that's the community engagement component of this. And there I want to recognize Sabrina Liu, who's here, and also Candace Taylor-Lucas, because those two are spearheading this. But what we've been able to do already is assemble a really extraordinary group of community advisory board members from organizations across Orange County who are really invested in child health and health equity. And then in collaboration with Dan Cooper and the ICTS, we are holding community engagement studios with sort of 3 distinct groups of stakeholders. So the pediatricians who are doing the screening, allied healthcare professionals who are in the trenches and really doing the screening, and then also parents. And in those community engagement studios, we have important dialogues and really getting important information about perceptions of screening in primary care settings, potential barriers to the people who are participating in this, potential benefits. And then also we'll be discussing getting input from these various groups on their feelings and thoughts about the science of AIM 2. So I might turn it back over to Tally.
Thank you. So the first aim is really to test in reality, in the real world, if unpredictable sensory signals from the environment are important. The second one, which is just as ambitious, is, okay, so we have 2 kids who seem to have exactly the same type of parameters. We have Johnny and Julia, and Johnny is going to be just fine, and Julia is going to be a train wreck in terms of mental health. Can we predict at the individual level early enough, so we can do something about it, that we can at least recognize that a child is at risk and give them the best we have? So for that, we aim to recruit 120 mothers and infants and really have the mother agree to obtain saliva, namely salivary and buccal swab cells, so that we can actually look at the DNA methylation. And we'll use intra-individual DNA methylation to identify individually predictive markers for neurodevelopmental impact. Now, I'm sure everybody in the audience is saying, intra-individual DNA methylomics, what the heck are you talking about? So let me just explain that a little bit. Again, the goal here is really to predict outcome for an individual child in the context of that child's exposure, not only to unpredictability, but actually to any kind of early life adversity. And how can we do that? Well, ideally, we would like to go into the brain and say, aha, there's a signature here, and that particular reward striatum, medial prefrontal cortex, whatever we've heard from Diego, and what we're doing in our own lab is impacted. But that is in reality not possible. So we would really looking for a peripheral predictive marker, not necessarily a biomarker, but something that tells us this child has been changed, this child has been impacted, where the other ones is not. Now, what's available to us peripherally is DNA. We all have our DNA. We all have many, many different kinds of DNA, and the variance here is unbelievable. And one thing that an environment can do is actually change the DNA by putting a variety of molecules on it. One of them is methylation. And there's a whole science of methylation, may or may not influence gene expression. It probably does. We don't really know if these genes will be important for the brain or not. We don't think that's important. But the question is, can we simply identify a signature in terms of a specific methylation pattern or anything that will happen to methylation in that individual's DNA, we'll call it a methylome, if you will, that will be a predictor, a presager, a marker, a predictive marker of what the environment did to the child. Now, this is not a new idea. I'm sure everybody in the room has heard of it. And in fact, last I went to the literature, there are about 550 studies to try to do the same thing, but they didn't really do very well. Why? Because humans are incredibly different, and everybody's DNA is different, and everybody's methylation prone cytosines are different. So it's incredibly difficult to get over the variance. You need Manhattan plots, you need maybe 100,000 people, and you may be able to get a correlation, but we don't want a correlation. We want to be able to predict for an individual child. So we thought we'd try to overcome that variance by something very simple. Rather than comparing one individual to 150 million humans, we'll take the same individual and compare them to themselves. And we tested it to give a test run in rats, and this is how it works. So we take rat A, Johnny, and we take a buccal swab sample on postnatal day 2. We then randomly assign that rat to either a comfortable existence, which I think is somewhere here, or to an adverse existence, and take a second sample from the same individual at postnatal day 10. In humans, what we're actually doing is taking one at about the 10th day of life, and then after they go through a year of life, and Laura's team, and together with Chuck and the rest of the team, really knows a lot about what happened during that first year of life. We actually take a second sample and look at the delta. What is the change in that individual between sample 2 and sample 1? And again, in our animal model, we were able to really come up to some very interesting conclusion. When we just look in the usual way at all of the samples, and ignore that, it's the heat map, or except for the cognoscenti follow, you can look at it better than I do. But essentially, you have a very clear ability using principal component analysis to tell the old samples from the young samples. Age influences methylation, and that is something that we can see when we simply compare samples. But if we try to look at, can we see any prediction of whether this individual had a good life or bad life, if you will, that is impossible. Conversely, if we do the other type of analysis, and I would like to absolutely acknowledge here Ali Motazavi and Hal Stern and the students who were involved in this, because then Mandy Jiang is the first author here, and Noriko Kamei and many others, this really sort of allowed us to go into the human. What you see here, that doing the same type of analysis, but focusing on the difference in the same individual, allows you to separate out those that had limited bedding and nesting, i.e. resource abnormality, and the very aberrant maternal care that goes with it, maternal behavior, and those that didn't. And at least in rodents, this is highly predictive, because as I've shown you at the beginning of the talk, the rodents, mice, and rats that have been through the adverse experience have these cognitive and emotional-like deficits that we talked about in a sex-specific manner. So the question is this going to work in infants? Well, we're fortunate enough to have begun to implement that in a Conte Center cohort that Alicia Davis and Laura Glynn have been collecting now for, I think, about 7 or 8 years. And these infants are now providing us with, if you will, a pilot set of samples, more than a pilot. We're also in the process of recruiting for the California initiative, but we can actually make progress in analyzing the data that we already have and using that to really guide how we're going to look at the California initiative. So again, what we have now is 115 infants, which means 2 samples each. And we've run them through a very complex set of pipelines. And again, I won't bore you with the details. But what is emerging now is, and I'll show you a little bit of these data, There is correlation with unpredictability. There is an emerging correlation with outcome and emergent genes that tell us what are the key changes, at least in the periphery, that may allow us to derive an impact score. So let me just give you some titillating early results that Annabelle Short and Ali and Hal have been involved with in the past few weeks. First, if you look at mean entropy rate, that does contribute to delta methylation. Again, we're interested in entropy rate. We'll be looking also at other types of adversities in the California Initiative cohort. But the idea is that in addition to anything else that may drive delta methylation, the difference in methylation in an individual mean entropy rate, namely unpredictability, is a predictor. If you focus on significant methylation changes and try to come up with something that may be an impact score, you actually get much better correlation, as you can see here. These are 241 sites that are significantly influenced here. In terms of looking at outcome, Laura has shown you this very important cognitive control or executive control or delayed gratification, which is a transdiagnostic, important predictor of how an individual will do throughout life. We're just beginning to show and to find that if you divide individuals again into those who don't wait or wait less than a minute and those who do wait, and then you look at the impact score, there seems to be a relation here that is emerging and actually is beginning to distinguish those that have had, if you will, an effect of the environment on the DNA methylome now are also those that maybe cannot wait or do not have cognitive control of the behavior even very, very early in life. And of course, these are just initial titillating data suggesting that what we are aiming to do with the California Initiative is feasible. So we've taken you through a tour to try to suggest that we really would like to know and improve the outcome of these infants in California and actually throughout the world, and really see if we can make a change in the impact of a variety of early life adversities on the outcome of the child. We are fortunate and privileged to have benefited from this initiative in the Office of Planning in the state of California. And what we're aiming to do is to really capitalize on the community, as Laura told you, on the community interest and passion, on team science and on precision medicine and the best cutting edge methods we can come up with to really improve the lot of the kids in Southern California. And of course, we have to acknowledge the team. We've tried to do a little bit of it throughout the talk, but this is a village, and a huge, amazing village that I'm fortunate and privileged to work with. The preclinical team with Ali's lab and mine, and of course with Hal Stern and his students. The Conte Center California Initiative team in Chapman that Laura runs. And again, these are some of the individuals from her team that absolutely do the work. The California Initiative team at UCI, again, the leadership of Hal Stern and Ali and Dan, working with Candace and Dave and Anton is unbelievable. And of course, the team at Chalk, led by Michael Weiss, Charlie Golden, and Louis, and I'm sorry, I always butcher his name, and Werner Pfeffer. And of course, we're really thankful to the state of California and the foresight and vision of the Office of Planning with Nadine Burke-Harris and Jillian, and the many other individual funding that allows that, and all of the people here. Thank you so much for listening. I think we're doing okay for time, and what I'd like to do is maybe invite Diego to come up here, and really we'll open it up for questions, and either come to the podium, yell, and if anybody is a remote, text James.
I gotta go first. Fine, I have a question. Thank you for the interesting talk, Dr. Pizzagalli. This one's for you. My name's David Keeter. So my question is about symptom clusters and the diagnosis of MDD, and the same applies, I think, to other psychiatric diagnoses. So I'm a data scientist, not a physician. But the way I read the DSM are there are potentially thousands of symptom clusters that will result in the same clinical diagnosis of MDD in this case, yet the biological mechanisms of those symptom clusters may be dramatically different, in my opinion. So, I mean, do you think that this is contributing to the slow progress in the field at understanding and developing treatments for MDD in particular? And then beyond that, what do you think we should be doing about that problem?
Can you hear me? Is it on? Okay, perfect. No, absolutely. So for me, that this heterogeneity and not paying attention to that, it's my opinion. One of the reasons that the progress has been relatively modest, and that's why early on we had really focused on what people describe potential intermediate phenotypes or sometimes endophenotypes, right? With the idea that they will be more tractable from a research perspective, but more important than that, perhaps they might be linked more lawfully to specific neurobiological abnormality, and potentially even linked to particular risk factor that we have been studying. And over the years we have done several studies where it's remarkable. You don't see any difference in the scale that we use, for example, right? In terms of depression severity, which again, even now depression severity scales, they aggregate across all these symptoms, right? Somebody comes out at the end as one number, right? On, let's say, the Hamilton Rating Depression Scale, for example. And we see that they basically have a specific depression severity, which is indistinguishable from other subgroups, right? And the approach that we've taken often in my lab and in other labs is that, well, let's step back and see, sometimes people describe it also as biotypes, right? Whether they are using machine learning and other types of approaches to see if neurobiologically they are, more homogeneous subgroup of patients characterized, by a specific, patterns. And that information could be very, very helpful to then decide how to treat them. So, this was just one study, but then we replicated in a different study, different cohort, using a different medication. I found it interesting that something that can be given, with our test, for example, or a relatively easy scan are expensive and require expertise to analyze. But I found it interesting that some of these markers allowed us now in 2 cohorts to identify patients that basically they really don't do well on SSRI. And arguably there is a world where we might be able to say with like a calculator, so to speak, right? That you input different variable and say, you know what? If you're interested in pharmacology, skip the SSRI. Which SSRI are typically 9 out of 10 times that somebody wants to start medication. And that's probably what the PCP will prescribe, right? So it's actually skip that and go to something different. Now, I've emphasized some of our work with pharmacology, but we have similar study also using, cognitive behavioral therapy, behavioral activation treatment for depression. Again, we're very excited by this new study looking at neurofeedback to try to, in quote, teach individuals to upregulate the reward system. At the end of the day, of course, we and others will need to replicate this, and but I am quite enthusiastic in the sense that, after many years of, ourselves and other people identifying these markers, I think we are at the verge of, again, we are implementing them. And it might not work out, but at least, the field is trying. Yeah.
Thank you. The microphones that also people can obtain. We have questions. Okay, I'm trying to see who's asking, but go for it. Ask.
Hi, I'm Kate Coleman.
Wonderful.
So one of the things I couldn't help but noticing across the 2 series of talks was that it seems like a lot of the action is coming from within person effects or within person sensitivity rather than between person effects or group level differences. And it's not my experience that the broader field thinks of things that way, or that like at review panels, people are willing to consider within person effects as rigorous as between group effects necessarily. And I guess I wondered if you guys could reflect on whether you think that that's true, my sense of that, and any strategies you have about dealing with that, or maybe like preaching the good word to people and trying to get more adoption of this within person, because that's always been my sense in my data, is that it's within person sensitivity and response bias, changes in methylomics over time. I mean, it seems like that's where a lot of the action is coming from in the data sets.
I'll answer it. All right. So, Kate, first of all, really good question. And one thing we can't do a priori, and I'm going to just say that we can't really figure out what study sections are going to do. That's just, I mean, crystal ball is as good as anything else. But let's sort of think about it. Where is the evidence? The evidence for 100 years has been really looking across groups. I mean, not at the individual level. And that's fine, and we want to acknowledge that. But now we're looking to the future, and I think we can build on that, show where the gaps are, as I was trying to do with this amazing Denisi work. And Denisi actually has been one of the pioneers. I mean, the people who wrote the paper showing that the group level does not apply to individual level are the people who led these huge Denadin, I'm sure you know the studies better than I do. They're the ones who pioneered the field. They're the ones who actually showed how important the studies are showing group level. But now they're trying to go in their own clinic and, or a lab and try to implement to an individual and realize that's not quite the case. I would go and give all these people the credit they deserve and then say, hey, this is the next challenge. These are the gaps. And I think the gaps really are, particularly with the theme here of precision medicine, is that we are, as humans, very, very different. It's not only true for humans. I mean, everybody in the lab knows there's some individual variability in rats and mice, but we can work around that or study it. But in human, everything is about the fact that we have a brain with 100 trillion neurons and Lord knows how many microglia and oligodendrocytes, and all the connections are different, and we absolutely need to embrace this complexity, or else we're not going to be able to move, the pendulum any further, or the needle. So I think that acknowledge, build, and move forward. I hope that's sort of helpful.
If I may, I also, I add, this is something that I didn't have a chance to emphasize, but many of us in the field also, of course, are moving toward very dense sampling of, maybe even fewer number of subjects, but very dense sampling, including using, for example, ecological momentary assessment, or different devices that basically can objectively and really passively characterize individuals, right? So in the study that we were just got funded by the Wellcome Leap to do, where we have this what they call the SMART trials. Also, we will use different type of gadgets, so to speak, just to passively assess them very densely. So somebody who has anhedonia, we should be seeing maybe fluctuation in their mood report throughout the day. And in fact, we have evidence that sometimes it's actually the variability within subjects, how people actually express negative affect, for example. And that standard deviation, that's really associated with maybe hyperactivity in the amygdala or other limbic regions when we, maybe expose them to stress in the MRI. So, I'm absolutely with you. I think that some relatively modest progress. It's also due to the insistence, which makes sense. Case control studies, right? And we've been very happy in the field for decades. Funding agency have been happy to fund that, but I think people get a bit impatient.
I was wondering, it seemed like in your studies you were finding that there was low dopamine transporters in depressed patients. Is that correct? So would you interpret that as being a homeostatic response to hypodopaminergia in these individuals?
That was exactly the hypothesis of the grant. And as I said, I don't personally believe that that's even the primary issues in MDD, but yeah, absolutely a downregulation to compensate for the low level of dopamine.
So do you think that maybe more dopaminergic stimulation might be appropriate as, it seems like the first line treatment right now is primarily serotonergic, but if it seems like you're suggesting that it's hypodopaminergia it seems like maybe there's not enough emphasis on consideration of dopaminergic stimulation, and perhaps.
Yeah. No, so the series of studies we have done really over the past 15 years has been trying to tackle that, right? And so what we have shown is that If we take healthy controls or healthy rats, and we give them now the opposite manipulation, which was a low doses of pramipexole. Now it's FDA approved for Parkinson, right? But interesting enough, again, at low doses has this very nice paradoxical effect. We believe is actually activating the presynaptic autoreceptor and inhibiting the cells. So you take healthy humans or healthy rats, and this has been replicated now a couple of times, and we give them this dopaminergic challenge. We make them anhedonic in our test. They look like people with depression. Now, if we do the opposite, we can potentiate this behavior. So yeah, we believe that, especially when anhedonia is a really important component, I think that, hypodopaminergic signaling is a key component. As we discussed in the paper of, in the discussion section of these two papers, when we wrote the grants and we wrote the paper in this way, of course, to be true to our hypothesis had been that people that have anhedonia, it's partially because they have low level of dopamine, and they will do really well on a dopaminergic drug. And we have tried both bupropion as well as pramipexole. And lo and behold, in both studies, we found that indeed the reward-related behavioral and neural markers were the only predictors of response to these drugs, but in the opposite direction that we have hypothesized. And we discussed this in the paper, say, it's really puzzling. They have potentially low level of dopamine, they display these anhedonic phenotypes, and yet it seems that their reward system has to be still a bit responsive, which is not what we had hypothesized. The hypothesis was the opposite in the grant. And so it seems that, and maybe from some people this makes sense, that some level of responsiveness has to be present in order to be able to respond, which is actually why we are particularly excited by this new neurofeedback study, because if that's the case, potentially, and we will start recruiting in July, we might be able to, in quote, teach individuals to upregulate that system, and then potentially they might be able to also respond to other treatments.
Okay, thank you.
Hi. Can I go next?
Of course.
Is that okay?
Yeah,
Fantastic.
go for it.
Autumn Ivy. And thank you for a wonderful morning session. Congratulations on the California Initiative Award. This is really important work. And so I have two questions, one for Dr. Glenn and one for Dr. Barham. For Dr. Glenn, I really think that the opportunity for you guys to do this work during this time of the pandemic and COVID-19 and how it's affected children disproportionately in certain communities more so than others. I'm wondering how you're thinking about how your parental unpredictability data is impacted by COVID-19 and its associated stressors in various communities. And I wonder how this shows up in your community engagement sessions. So wanted to hear a little bit about your thoughts on that. And then for Dr. Barham, also wonderful to just always hear about this translational work, and the Conte Center models this beautifully. And so thinking about DNA methylation from saliva and how we think about kind of this epigenetics, it's all epigenetic mechanisms, how we're thinking about how that DNA methylation may turn on or off genes. Are you wondering if this DNA methylation signature that you're seeing in saliva has anything to do with reflecting brain-based behaviors or stress responsiveness, mental health outcomes? And if you have any predictions with regard to the signatures of the DNA methylation, because I know in your titillating data, there was delta methylation, and then there was significant DNA methylation. So I wanted to hear a little bit more about that. Thank you.
So in terms of the CES and COVID, I suppose that could sort of organically emerge, but because of the science of these community engagement studios, it's guided discussion by a facilitator that's very focused on the issues at hand. So ACEs screening in primary care clinics and predictability also. So we'll see. I can't right now give you any feedback on that. And I don't even know, without specific questions in the discussion guides that we've created, which don't include COVID or pandemic related questions, I'm not sure how much we'll see there. I can, before I turn it over to Tally, just give you one bit of information that the center has generated regarding predictability and child mental health in the context of the pandemic, and also acknowledge, first of all, that of course the distribution of this in terms of stress and mental health is not even across different populations of children and individuals. But we have shown that despite all of these disruptions, among families where the family unit or the parents are able to maintain more consistency and routines, the mental health of those children is doing far better. So that may fit with intuition of people, but we've actually documented that in sort of preschool age children, and now that's been replicated in older kids. So kind of underscoring, even in the context of pandemic, the importance of predictability for child mental health.
Actually, Laura, you not quite give yourself the credit you do believe. So when COVID hit, it became so clear, Adam, that we have a huge elephant sitting here, and that's going to absolutely interfere, confound not only children's mental health, but also potentially the studies we're doing. So Laura and her team put together a questionnaire within 2 weeks when California shut down in May, and sent it to 500 kids and families? 200 and some odd. And particularly focused initially on preschoolers. Here I am extolling your glory. I mean, Laura, you should do it. Came up with really one of the first and critically important paper that's now cited by the not California Surgeon General, but a Surgeon General of the whole country in their volume on issues of mental health and COVID, and including depression. So there is a manifesto that came out, I think, 2 months ago, and cites Laura's work on that. And what fundamentally found is, yes, there is a huge mental health impact on preschoolers in this particular case, and now also adolescent, and I think there may be a poster this afternoon about that. But there is a huge impact on preschoolers, regardless of the socioeconomic status, and again, it's beautifully delineated what this mental health impact is. But the protective factor seemed to be predictability within the family and maintenance of normal routines. The paper came out in Neurobiology of Stress last year, and I really recommend it. So hopefully that's another way to address your question. Laura, what did I forget?
What's that?
I mean, what part did I forget?
I don't think you forgot any.
So kudos to Laura and the team. Yeah, this has really been impressive. And again, they... Now, to answer the question about methylomics, so I need to take a deep breath because this is a wasp's nest. First, yes, I think changes in DNA methylation are clearly associated with changes in gene expression. We are looking at a combination of white cells and epithelial cells in the mouth. And these cells can tell us a lot. And I'll tell you more about this in a minute. But before I get there, I have to say very explicitly, I don't think that the same thing needs to happen, quote unquote, in the brain. And because once we get into the brain, we need to go where in the brain, which types of cells, which, again, with single cell transcriptomics, there's so many different individuals. I honestly believe that what happens in the periphery, namely in the buccal swab cells, may be quite informative and parallel, similar, maybe even overlapping mechanism may go on in the brain, but that's not the purpose. Now, with this in mind now, I can really address your question. And we have a fair amount of information of what happened in the rodents because we've done this sort of a trial run, a pilot to see if this whole concept works in the rodents. And we found that, yeah, there's some very specific types of genes that emerged. They were particularly transcription factors. They were particularly cells that are involved in metabolism and response to stress and inflammation. So the whole thing kind of makes sense. And again, the idea is if methylation is reduced, there's more of the expression of these genes and what have you. We're beginning now, and I think Annabelle Short deserves all the glory here together with Hal Stern's group, to really see what kind of genes are involved. Clearly, some are more impacted than others. We're starting off with CPG sites and then working back to genes, and are really working hard to try to see if we can make any sense of which gene groups or clusters are really impacted, with the idea that that will give us clues in terms of what's going on. Again, not surprisingly, we're going to be paying attention to solute carriers and metabolic genes and, phosphorylation, potentially transcription factors, because, they may regulate whether a gene lives or dies or turns into, the cell lives or dies or maybe tries to turn into a more primitive cell. So this is something that's very much in the works, and hopefully we'll be able to share with you. So that may very well be a solid biological and meaningful marker, maybe even a predictive marker, but I will never aim to consider that that's exactly what happens in the brain, because it probably doesn't. Thank you.
Oh, hello, wonderful talk. And my question is for Dr. Pizzagalli. I'm a pharmacist and also I've done a lot of IT work. If you could elaborate more regarding the gene biomarkers, let's say, and I'm more interested in the pharmacological treatments. And you mentioned sertraline, one of the two drugs, and I'm assuming you're talking about following patients with CYP2, C19, metabolism, knowing their biomarkers. Is that how your study was performed? And then in recruitment, whether you actually saw a change, if you changed doses, if you offered alternative treatments based on poor metabolizers or not. So if you could elaborate, I would appreciate it.
Happy to do, certainly. Thank you for the question. So, so the study is, we just submitted the RB protocol, so we have not started yet, but we hope to do so soon. But basically, again, over the years, and especially in the context of a multi-site study, we were fortunate to participate in called the EMBARK study, where again, hundreds of patients across the nation were recruited and treated. We identified markers that predict response to sertraline and markers that predict response to bupropion, which again has a different mechanism of action. So the design will be that we believe we need probably assess about 180 patients using behavioral tests, clinical scales, but also imaging. And then the plan is to identify among these 180, 120, 60 we think will have the markers that we have linked to better response to bupropion, and half of them will then go on and receive bupropion, and the other half will receive sertraline, and vice versa for the sertraline group. So because this is potentially the first time this has been tried, right? We need to basically convince ourselves in the field that this guided treatment, overperforms. Our goal in terms of, the proposal and the RFA was basically they know the Welcome Leap came out with this request for proposal, acknowledging that the presence of anhedonia is associated with poor response to treatment. And the goal is to try to double their response to therapy, to treatment. And so obviously the hypothesis that those receiving the intended treatment will do much better compared to the others. We will also collect DNA and other epigenetic and other markers, and that would be a bit more exploratory. And incidentally, again, maybe also for those interested, many of us have emphasized team science. The reason that we have not started yet is that the founders, in this case, the Welcome Leap, really had the wisdom of awarding 12 teams around the world. And they said, before you start, we'd like that you try to harmonize as much as possible. So we spent months discussing among each other which scale to use, which behavioral test to use, which acquisition protocol to use, which epigenetic protocol to use, and so on, biomarkers, and we're done. So we are now, all the sites will start using the same, physical protocol. Sometimes answering different questions, of course. Everybody will be pursuing something different, but really, a wonderful example of how, scientists are eager to, collaborate, to join forces, and all the data then will be sent to a central repository at Vanderbilt, where, they will do a lot of machine learning and artificial intelligence to really try to develop markers of anhedonia.
Thank you. We'll probably stop the session now, although I'm sure there are many additional questions in here. And what we'll do is we'll take a break until 12:10. Please then come to the poster area for the poster tours, and then we'll have lunch at 1 o'clock. And this will get back on schedule. And I'd like to really thank you in the audience, both here and remotely, for the terrific enthusiasm and for listening. Thank you.
sorry, that I